All right, folks, here we go. This is 14-2. This is the meninges and cerebral spinal fluid. I mean, you saw it a little bit in the introduction to the brain, but now we're going to talk some more detail about the three layers of meninges and the cerebral spinal fluid that exist underneath one of the layers. Well, directly underneath one of the layers, actually exists underneath two of the layers. So let's get going on this. First of all, you have three layers of meninges, and when you look up here, you can see the three layers. You can see the dura mater right here. Right here's the dura mater. You can see the arachnoid mater right here, and you can see the pia mater right here. And this is the brain. All right. Now, I know it shows you the dura mater is splitting, and it certainly does, but let's come back to that. This is the dura mater. It's a real tough outer layer, as you'll see when you look at a sheep brain in lab. This is the arachnoid mater right here, and underneath the arachnoid mater is the subarachnoid space right here. This is where the cerebral spinal fluid bathes the brain. So the cerebral spinal fluid is directly underneath the arachnoid mater and on top of the pia mater. That's where the cerebral spinal fluid is. Now, the dura mater, in some places of the brain, the dura mater splits. They're still both dura mater though. The layer that's closest to the arachnoid is called the meningeal layer because it's closer to the other the meninx called arachnoid mater. The layer that stays tightly adhered to the bone is called the periosteal layer because periosteum is the surface of a bone. So my dura mater splits and I have the periosteal dura and the meningeal dura. And what's in between that is called the dural sinus. This is venous blood in here. And this venous blood from the dural sinus is going to drain into the internal jugular vein. So really the venous blood of the brain drains into the dural venous sinuses. They're called dural venous sinuses. And then the blood from the dural venous sinuses drains into the internal jugular vein. And then back to the heart, of course. So those are the three meninges. The big thing here being, the two big things being CSF is in here in the subarachnoid space, and venous blood is in the dural venous sinuses. All right, that's it for right now. That's it for right now. Let's go on to some more details. I want you to look at some folds of the dura mater. All right, so this first fold of the dura mater right here, this is called the falx cerebri. This folds down between the hemispheres of the brain. The brain, as you probably know, has a right and a left hemisphere. And this falx cerebri is a fold of the dura mater that delves in between these hemispheres. Matter of fact, up here in the front, this falx cerebri hooks to the rooster's comb, the crista galli of the ethmoid bone, if you recall that from Bio 203. There's a crista galli of the ethmoid bone. And the falx cerebri hooks to that. Notice the dural venous sinuses here, 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 up here. These are dural venous sinuses. Notice they do have some names. Superior sagittal sinus, transverse sinus. So these dural venous sinuses are all eventually going to dump into the internal jugular vein. All right, now we have another fold of the dura mater, and it's called tentorium cerebelli, and it's right here. This tentorium cerebelli is a fold of the dura mater that folds into the transverse fissure, which separates the cerebrum from the cerebellum. So this dura mater folds in between the cerebrum and the cerebellum. So all of this up here would be cerebrum, and there here's your cerebellum. And this tentorium cerebelli folds in between the two and separates the cerebrum from the cerebellum. And then lastly, I have this fold right here. And it's hard to, I'm going to tell you what it does. It's hard to see from this viewpoint. But this is called the falx cerebelli. And like the cerebrum, the cerebellum has two hemispheres. and they're connected by a vermis, but that's neither here nor there. It does have two hemispheres. And this falx cerebelli is a fold of the dura mater that folds in between the two hemispheres of the cerebellum. So right here is the falx cerebelli. So these are folds of the dura mater, and you can actually see the tentorium cerebelli right here, a little bit of it, by the way, folding in between those. You really can't see the falx cerebri because I can't see in between the two hemispheres, so I can't really see that. And by the way, I can't see the falx cerebelli in this picture over here because I can't see in between the two hemispheres of the cerebellum either. So there we go. That's the meninges and their folds. Now, as I mentioned in the last lecture, you make about 500 mils of cerebral spinal fluid a day, but at any given time you only have between 150 to 180 milliliters. So if you make 500 mils per day, but at one given time you have 150 to 180 mils volume, how can that be? And the answer is that you're always reabsorbing the cerebral spinal fluid. So you're always making it and you're always reabsorbing it. That's the answer to how could these two things be true. So let's start with making the cerebral spinal fluid. First of all, the cerebral spinal fluid is made in the ventricles. So this right here is it's one of the ventricles. We're calling it the third ventricle. It doesn't really matter which one you called it. All right, so here's the third ventricle. Here's some capillaries, some capillary blood right here. Now, what the cerebral spinal fluid really is a filtrate of our plasma. Because what happens is these ependymal cells right here, these are glial cells that you learned previously. They're ependymal cells. These ependymal cells absorb some plasma, modify it, and secrete it into the ventricle. So cerebral spinal fluid is a filtrate of plasma. Now, it's not identical to plasma. It doesn't have the same amount of protein. It doesn't have the same amount of glucose. It doesn't have the same amount of white blood cells, et cetera, et cetera, et cetera. So it is a filtrate of the plasma. It is modified plasma, and these ependymal cells are the cells that modify it. Now, I want to point something out here. Look at these tight junctions. These tight junctions right here. This is part of the blood-brain barrier. Anything that goes that gets to the brain, like this, that gets into the cerebral spinal fluid. If it's in the cerebral spinal fluid, by the way, it has free access to the brain. Look at right here. This is CSF. Look at the free access to the brain I have. Absolutely free access. So we better kind of control what gets into the cerebral spinal fluid. And indeed we do. These ependymal cells have tight junctions between them, and they control what gets into the cerebral spinal fluid. They maintain this blood-brain barrier. Now, I will tell you this, that this is one of the areas where the blood-brain barrier is not as tight. There's 4 areas where the blood-brain barrier isn't as strong a barrier as the rest of the brain. And the place where we make cerebral spinal fluid is one of them. They're called choroid plexuses. Matter of fact, the choroid plexus, this word right here, a choroid plexus is the blood vessel and the ependymal cells acting in conjunction with each other to make CSF. That's what a choroid plexus is. So there's a choroid plexus associated with all the ventricles. So there's choroid plexuses in the 2 lateral ventricles. There's choroid plexuses in the 3rd ventricle. There's choroid plexuses in the 4th ventricle. And what do the choroid plexuses do? They produce CSF and they remove waste from the old CSF. This is what they do. They remove waste and they produce new CSF. All right. This right here is a list for you the 4 main breaks in the blood-brain barrier. BBB stands for blood-brain barrier. First of all, the 3 of them are because of hormonal production. These 3 are because of hormones. The last one is because of making CSF. All right, so number 1, the hypothalamus has a place where there's a break in the blood-brain barrier because the hypothalamus makes releasing hormones and inhibiting hormones that have to be delivered to the pituitary via the bloodstream. So there's a break in the blood-brain barrier there because the hypothalamus part of the brain makes these releasing and inhibiting hormones that need access to the blood. There's a break in the pituitary gland because what happens is the hypothalamus makes two hormones that get delivered to the posterior pituitary gland. But you don't need a break in the blood-brain barrier for that delivery because the hormones from the hypothalamus that get delivered to the posterior pituitary, those hormones are antidiuretic hormone and oxytocin. They actually get delivered to the posterior pituitary via neurons. However, the posterior pituitary has to be able to secrete those two hormones into the bloodstream. So there's a break in the blood-brain barrier at the posterior pituitary because those two hormones, ADH and oxytocin, need access to the bloodstream. The next hormone, melatonin from the pineal gland. The pineal gland is part of the epithalamus, and the pineal gland makes melatonin, and melatonin needs access to the bloodstream. Okay, so those are 3 of the 4 breaks. The last break is in the choroid plexuses, which are blood vessels and ependymal cells. And this is because the ependymal cells need to have access to the blood plasma so it can modify it and secrete it into the ventricle as CSF. So there's a break in the blood-brain barrier, or a weak in the blood-brain barrier at each of the choroid plexuses. All right. Those are the 4 breaks in the blood-brain barrier. Let's talk about the circulation of the cerebrospinal fluid. So by the way, there's a septum pellucidum right here. Right here is a septum pellucidum, a thin membrane. And that's why this choroid plexus is a light red, because it's trying to give you the impression that you're looking through the septum pellucidum. You can't see the choroid plexus all that clearly. But this choroid plexus right here of the third ventricle, you're not looking through a membrane, so it's a darker red. It doesn't look like it's trying to make it look like you're looking through a membrane. And this is another choroid plexus down here in the fourth ventricle. So the lateral ventricle, both lateral ventricles have choroid plexuses. The third ventricle has a choroid plexus, and the fourth ventricle has a choroid plexus. This is where CSF is made. Now what happens? Let's just start from the lateral, the most superior ventricle. CSF is made in the lateral ventricle, and it flows through the interventricular foramen into the third ventricle. Now CSF is also made in the third ventricle. All of that CSF flows through the cerebral aqueduct into the fourth ventricle. Now the fourth ventricle also makes cerebrospinal fluid. All of that cerebrospinal fluid flows out of my apertures. There's 2 lateral apertures and 1 median aperture, and it goes into this subarachnoid space. This is the subarachnoid space right here. Subarachnoid space. I'm not going to stay in it all that well if I keep going. So all of this is subarachnoid space. So in other words, the cerebrospinal fluid bathes the brain and spinal cord. It bathes all of it. In addition, the fourth ventricle is continuous with the central canal of the spinal cord, and the central canal is filled with cerebrospinal fluid, although that probably dries up and collapses as an adult. But as a child, your central canal is wide open and filled with cerebrospinal fluid. So there you go. Now what I want to do is I want to zoom in on these things right here. These are called arachnoid granulations. And this is the dural venous sinus right here. So I want to zoom in where my subarachnoid space, let me erase what I circled so you can see it, where my subarachnoid space has these arachnoid granulations dipping into the dural venous sinus. Because what's going to happen right here is you're going to reabsorb cerebrospinal fluid back into your bloodstream. That's what's going to happen there. So let's zoom in on that. All right, here's the arachnoid granulation right here. So you have the arachnoid granulation. You have arachnoid villi and arachnoid trabeculae right here. All right. And right here, cerebrospinal fluid is going to be reabsorbed back into the bloodstream, right back in the dural venous sinus. So all along these venous sinuses, these dural venous sinuses, I have these arachnoid granulations. And the arachnoid granulations are where CSF is reabsorbed. And that explains how we can make 500 mils a day, but at any given time only have 150 milliliters of it, because we're constantly reabsorbing it and constantly making it. So a snapshot of our volume of CSF is 150 mils, but overall we make 500 mils a day. By the way, when you do spinal taps and you pull out CSF, an adult can give probably about 10 mils of CSF, and the lab takes 10 mils and divvies it up between microbiology and clinical chemistry and hematology, and every section does its test on the cerebral spinal fluid to see if there's bacteria in it, or abnormal cells, or abnormal proteins, which sometimes mean a lot of antibodies fighting off infection, or it could be abnormal or abnormally low glucose in there, which could mean there's bacteria growing in there metabolizing the glucose, etc., etc. But with a child, especially a young child, toddlers, and even I say prepubescent kids, you're only allowed to take maybe an ml or maybe 2 mLs if it's a bigger child, because they don't have 150 mils of cerebral spinal fluid. It's very difficult for the lab to divide up and run all the tests on spinal taps from kids. We do it. We have certain procedures that preserve, and maybe share, which we don't normally like to do, and stuff like that. So you can't take as much CSF from a spinal tap from a child as you can from an adult. All right, so that's the reabsorption of cerebral spinal fluid.
Within the skull and vertebral column, the brain and spinal cord are protected by 3 layers of connective tissue, collectively called meninges. From superficial to deep, the meninges consist of the dura mater, arachnoid mater, and pia mater. Within the cranial cavity, the dura mater is composed of 2 sublayers. The outer portion of the dura, the periosteal layer, is tightly adherent to the inner surface of the skull. The inner portion of the dura is called the meningeal layer. In some areas, these sublayers separate, and the meningeal layer folds inward to divide and support the major parts of the brain. Dural venous sinuses are formed at sites where the periosteal and meningeal layers of the dura split. These sinuses collect the venous blood from the brain. Internal to the dura mater is the arachnoid mater. The arachnoid is composed of a thin, transparent membrane that is normally in direct contact with the overlying dura. The arachnoid also includes web-like extensions that stretch inward to reach the pia mater. The space between the arachnoid and the pia mater is called the subarachnoid space and is normally filled with cerebrospinal fluid. The pia mater is a delicate layer of connective tissue. It is located directly on the surface of the brain and spinal cord and follows all of their surface contours.
Okay, the key thing she said there about the pia mater was that it's actually difficult to separate the pia mater from the brain. It dips into every sulci, it adheres to every gyri, it dips into every fissure. It follows the contours of the brain very tightly, and when you do the sheep brain, you'll see the dura mater is easy. You might even see the arachnoid mater relatively easy, but the pia mater is tightly adhered to the brain, and it is what gives the brain that shiny appearance you're going to see. And you probably could tease it away, but you're not going to be able to tease it away and get an intact membranous structure on your forceps. It'll peel away in pieces. It is tightly adhered to it.
Cerebrospinal fluid, or CSF, circulates freely through the ventricles of the brain, the central canal of the spinal cord, and the subarachnoid space. It is produced in each of the 4 ventricles by the choroid plexus, a structure that consists of specialized ependymal cells, their associated blood vessels, and connective tissues. The endothelial cells of blood vessels within the choroid plexus and ependymal cells are joined by tight junctions and collectively form the blood-CSF barrier, a portion of the blood-brain barrier. The selective permeability of this barrier controls what molecules are able to reach the cells of the brain. We can trace the flow of CSF starting in the lateral ventricles. From the lateral ventricles, CSF flows through the interventricular foramina to the 3rd ventricle. From here, the CSF enters the cerebral aqueduct and travels to the 4th ventricle. Once in the fourth ventricle, the fluid enters the subarachnoid space around the brain and spinal cord by either passing through the two lateral apertures in the walls of the fourth ventricle or through the single median aperture located in the roof of the fourth ventricle. A portion of the CSF also enters the central canal of the spinal cord. Accumulations of arachnoid villi, called arachnoid granulations, project into a venous channel within the dura mater known as the superior sagittal sinus. CSF is recycled into the venous part of the bloodstream at the arachnoid granulations. The turnover rate for CSF, from its production at the choroid plexus to its reabsorption into the superior sagittal sinus, is approximately 0.4 milliliters per minute.
All right, folks. Well, there you go. That's CSF flow that we've already done on a static picture, and now you've seen the CSF flow from that video. All right.